It's a great pleasure to have Madan Rao give the talk. Madan is at the Raman Institute and NCBS, and that's what he'll talk about.
Bullseye instead of Spindle, right? Okay, so
the answer changed in the
that's right. Yeah. So thanks for being here and not wanting to climb the endless ladder or limitless ladder. So, oh, oops. So this is what I'll talk about. I'll talk about our ongoing work for the last 10 years on looking at patterning of molecules on the cell surface, both in mammalian cells and in yeast cells. And the bottom line is that this patterning is orchestrated by the coupling of the molecules to the cortical actin below. I recognize that a lot of you are not biologists, so I'll explain all these terms and I'll actually show you a cartoon of the cell. We try to understand this patterning from physics, new physics principles, and which come under the purview of active hydrodynamic equations. So I'll be talking about that briefly, and I'll show how these hydrodynamic equations gives rise to new phases of cortical actin on the cell surface, which in turn gives rise to patterning, nanoscale patterning and larger scale patterning of cell surface molecules. Yeah. Okay. I'll also show how these active stochasticity in this patterning of cortical actin on cell surface gives rise to huge number fluctuations of cell surface molecules. And I'll show how this patterning gives rise to an interesting spatiotemporal regulation of chemical reactions happening on the cell surface. I might talk of some of this. Oh yeah, this has to do with the Bullseye pattern, and this has to do with the rings. Okay, so this is a famous recent experiment done in David Weitz's group, which really shows very beautifully the unusual nature of cellular material. And so this is both cellular extracts and reconstitutions of these semi-flexible polymers called actin, cross-linkers, and motors with energy, with fuel. When these systems are inserted in narrow capillaries, which are bounded on either end by oil droplets, then the contractile forces which are inherent in this material make this material inside the capillary peel away or from the walls of the capillary and constrict like so. Okay, accompanying this constriction, there's a deformation of the oil droplet because it's attached to it, and by studying the deformation of this oil droplet, they can compute the contractile force, which is around a micronewton, which works out to around 100 piconewton per actin filament. I think it's reasonable to say that while this experiment is of course beautiful and they got a very precise number here, I think the details of the physics of how this sort of contraction happens is not well understood. So here's a cartoon of the cell. I just put this so that just to be internally self-contained. Here is the cell wall, and just below the cell wall is this thick meshwork of cortical actin. Okay? That's around 1,000 angstroms in thickness, and we'll be studying the organization of molecules on this cell wall, okay, on the plasma membrane of the cell. And the idea is that the local organization of molecules, the specific class of molecules that I'll be looking at, is governed not so much by protein-protein interaction, but by its coupling to this cortical actin below. Okay? And the forces that this cortical actin below the membrane apply are akin to the forces that we see here. Okay? And these forces that this cortical actin applies on the molecules and on the membrane here gives rise to currents which lead to clustering of molecules, localization, and transport. Okay, so as I said, we're going to focus on that little region here, and if you blow it up, that's how it looks like. That's the cell surface, the plasma membrane of the cell, and just beneath that is this dense meshwork of actin. Okay, And what we're going to discuss is how organization of molecules on the cell surface are controlled by the dynamics of this mesh of actin. Okay. Yeah, so here are real experimental pictures. This is the nanoscale patterning of actin, of the cell surface molecule called Gpi proteins. These are proteins with a lipid anchor, which allows them to sit on the outer leaflet of the plasma membrane. We'll show what we have discovered here. This is in very intimate collaboration with Jitu Mayer's lab at NCBS. What we discovered here is that these Gpi-anchored proteins organize as nanoclusters and reside on the cell surface. And this nanoclustering and the dynamics of these clusters is is controlled by the cortical actin below. How do I do that? This here is the ring that I talked about. This is an actin ring, which forms on the surface of yeast cells. And these have a certain mutation, which we'll talk about later, makes these actin rings slip and slide along the surface of the cell. Okay? Here is the Bullseye pattern. This is not a movie. Okay? So this is the Bullseye pattern that I talked about. This is again on the surface of T cells. The scales here are of the order of tens of nanometers, whereas here they are of the order of microns or more. Okay? And in red here are T cell receptors, in green are a co-receptor called ICAM. Okay? And it forms these Bullseye patterns. Again, these patterns are formed because of its coupling to cortical actin below. Okay. That's the group that did all the work, Ron Whale and Mike Dustin's lab. Okay. So we'll first talk about this, then that, and if I have time, I'll talk about that. Okay. So here's a Gpi-anchored protein. This is the protein head, which is tethered by this motif to a lipid anchor, to a lipid tail, and there sits on the upper leaflet of the cell surface. Okay? At optical resolution, it seems to be uniformly distributed. However, at very fine spatial resolution, using FRET, which Jayant Udgaonkar talked about today, we find that these Gpi-anchored proteins are distributed as monomers and these tight nanoclusters. Okay? 4 to 5 molecules in a nanocluster. And this technique, which is FRET, that is, we observe this distribution of nanoclusters and monomers, both by FRET and by a recent work by Garcia Parajo, which uses ENSOM, observes the same kind of architecture. Other work using EM also sees the same kind of architecture. So many techniques, independent techniques, have observed that these class of molecules form nanoclusters and monomers. And what is interesting, furthermore, is that these nanoclusters are formed because of its coupling to actin. Okay? I won't go through the detailed experiments that show this. It's all given in gory detail in these references. Suffice to say that these nanoclusters are not stationary or not static. They break and they reform. And the time scale of breakup is around 0.1 seconds. Moreover, they are associated with very particular regions of the cell surface. So they're predominantly in flatter regions of the cell surface, where the cortical actin is tangentially disposed to the membrane, and absent from these pokey regions, where the cortical actin below is poking into the membrane. Okay? So that's the other picture here. They're relatively absent from these microvilli and from cell edges, called lamellipodia. Okay? However, they are very highly enriched in the flat regions of the cell surface, where the actin looks like this carpet here. Okay? So the bottom line now that I'm trying to convey is that these nanoclusters are dynamic, and they are controlled by the dynamics of this cortical actin below. The cortical actin has an architecture which is tangential to the membrane and not normal to the membrane. Okay? So the cortical actin applies tangential stresses on the membrane. And the spatial variation of the tangential stresses can give rise to currents and therefore possibly to clustering. Okay. So if there are any questions, please do stop and ask me. Since this is the last talk, we have unlimited time. Okay. So now the idea is that since a whole lot of these experiments suggest that this clustering of molecules on the cell surface are affected by the activity of cortical actin below, we'll need to have a dynamical theory for cortical actin and then ask how do the cell surface molecules couple to this dynamics of the cortical actin. Okay? So we'll first see how do we write down or understand the dynamics of the cortical actin just below the plasma membrane. Okay. So we realize now that cortical actin is an active filament, as I showed in the first slide. Cortical actin in association with myosin, which is a motor, and ATP. These filaments are active, which means that they consume ATP. There's some local cyclical conformational change, and as a result, there's a movement of this filament motor complex. Okay? The motor here pulls on the filaments like so and like so. Okay? The dynamics of suspension of such filaments in a fluid has been worked out by several groups. One of the prime pushers or pullers of this field is Sriram here. And the whole lot of people, the Curie group and people from Syracuse and so on and so forth. So many people have worked out the dynamics of a suspension of filaments like so. Luckily, all the answers seem to be the same, which means there must be something right. So, but the approaches that each of these groups takes are different. Okay? So now, what I'd like to say is that if you consider a collection of these objects in a fluid, as a result of their movement, they apply stresses in its neighborhood, and these are the kinds of forces that they apply or stresses that they apply in the medium. One is that this actin motor complex is a polar filament. Okay, it's polar, so which means that there's a difference in the head and the tail. Okay, and which means that these molecules, one of the properties of these filaments is that they can polymerize more at one end and less at the other, which allows them to move in a particular direction. So, for instance, they can polymerize more there and depolymerize there, thereby move along this direction. Okay? And therefore, you can see that if it polymerizes more here and is capped there, then it can apply forces at this end. Okay? The other thing is that these filaments are attached to motors, and the motors consume ATP, and the motors move on the filaments, thereby moving the filaments themselves with respect to the solvent, and therefore can apply local stresses that way. So the two ways in which the motor filament complex applies stresses in the medium. One is due to treadmilling, the differential way in which polymerization and depolymerization happens, and actomyosin contractility, the forces generated by myosin moving on actin. Okay? Okay. So given this, if you have a collection of filaments like that, this is just a cartoon of this cartoon. Okay, if you have a collection of filaments like this, then as we said, it moves with respect to the solvent because it's polar. It also applies local stresses in the medium. And the stresses to lowest approximation can be seen to is just proportional to the local orientation of the filament. The local orientation is constructed from this second rank tensor constructed from this vector n. So n is the polarization of this filament. And you can construct a second rank tensor from this. And by just symmetry arguments, the stress tensor is proportional to this Qij, which is the orientation tensor. Okay? So what we'll be doing now is to write down, in order to study the dynamics of a collection of the filaments, you'll have to study what is the dynamics of the concentration of these filaments and the local orientation. Okay? So here are the set of equations that have been written for this. We look at the dynamics of this vector orientation. Remember, the filaments are polar. So it's described by a local vector n. Write down the dynamics of n. You write down the dynamics of the filament concentration c. And you write down the dynamics of the hydrodynamic velocity, the velocity of the fluid solvent plus the filament, okay, v. The equation for the hydrodynamic velocity is simple because it's a highly viscous medium. Force balance is valid. The divergence of stress is 0. The total stress is a sum of 3 parts, the dissipative part, which is just a viscosity into the strain rate, an order parameter part, which comes from the local orientation of these filaments, and an active part, which, as I said, is proportional to the orientation of the filament. Okay? So it's with the proportionality constant W, which is a measure of the activity. So that accounts for the force balance or the equations for the hydrodynamic velocity. The dynamics of the concentration field, if you assume that there's no polymerization, depolymerization, then the dynamics of the concentration field is conserved, and therefore it's just the divergence of a current. The current has two parts. One is the standard diffusion, and the second is an advection part. This is a purely active contribution and reflects the fact that these filaments could move because of differential polymerization from here and there, or because of actin contractility. Okay? So that accounts for this equation. This equation is the dynamics for how the orientation changes as a function of space and time. On the right are standard terms which would appear in any polar nematohydrodynamics. However, there's an additional term here which I'll come to later. So this just says that the dynamics of the orientation comes from a part that, okay, this says that n has a non-zero. This allows for n, or the magnitude of n, to have a non-zero velocity. That is, it moves. The filaments move. Okay, and so the local n here would change because of that movement. Okay, this would say that if two filaments are pointing in some random directions, then they would like to be parallel to each other. Okay, this I'll come to this term later, and this is a new active term. Which says that the filaments, if there are two filaments sitting like so, then this filament orients relative to this filament. Okay, so this filament advects itself. Oops. Yeah. Okay. Now, with this set of equations, these set of equations have been applied to various cellular contexts and have been quite successful in qualitatively addressing some of the features, though not quantitatively as yet. We'll try to use these set of equations to address the clustering and the dynamics of these specific class of molecules on the cell surface. And so, in order to model the cell surface in terms of its actin orientation, this is how we proceed. Remember, we said that the dynamics of clustering of these cell surface molecules is around 0.1 seconds. And if they are to be controlled by the dynamics of actin, then the dynamics of actin should be of that same order. So we assume that there are fast-moving, shorter filaments of actin just beneath the plasma membrane, in conjunction with these long cross-linked stationary actin filaments, which are seen in any standard EM picture of the cell surface. Okay? These filaments are dynamic, and they are described by a local concentration field and an orientation. And when they are next to this stationary filaments, they are preferentially anchored. Okay? So here again is the dynamics of the orientation and the concentration. Again, and these are the molecular, these are sort of the cartoons which describe each of these terms. For instance, this term, which I didn't describe to you earlier, has to do with the fact that if there are more filaments there, then there'll be more myosin motors, which will then result in pulling this filament towards that. Okay? So this says that if zeta is positive, it will say that it likes to align n and pull it along regions like this. So as I said in the earlier slide, the rate of change of the orientation is proportional to this active advection term, plus this part, which likes to make filaments parallel to each other, and this part, which I'll call the active myosin contractility, which likes to which pulls filaments in regions of higher filament concentration. In addition to all this, there is an active noise term whose variance is not kT, but some active temperature. The concentration equation, as I said, is conserved, so the time derivative of that is the divergence of a current, and the current has a diffusion part and an advection part and a noise. Okay, now what we'll do is, the strategy is that we shut off the noise and ask what are the steady states of this equation, then we turn on a noise and ask what is the dynamics in the steady state. And so the steady states of this equation are the following. If I plot the contractility in the x-axis and the relative alignment in the y-axis, yeah, yeah. No, it doesn't. So I'm just calling the variance of this noise an active temperature, for lack of a better word. But it has to do with the stochasticity of myosin binding and unbinding onto it. So it has to do with some chemical kinetics of how myosin binds to. Okay, so if I plot the contractility versus the alignment, for small contractility, you get this Asters street phase. Okay, you find that you have a low density of Asters, and in between you have filaments which are parallel to each other. Okay, as you increase the contractility, you find that this scale becomes comparable to that scale, and the system locks up into a lattice of Asters. Okay, so in increasing the contractility, you go from a moving phase, because remember, if filaments are pointing in that direction, the orientation of the filament is equivalent to the relative velocity of the filament with respect to the solvent. So it will move. So these phases are moving phases, whereas these phases, where there's no net orientation, is a stationary phase. So in going from here to there, wherever you increase the contractility, you go from a moving Aster street phase to a stationary Aster lattice phase. You can plot the net velocity of this object as a function of this contractility, and this is what it shows. Close to that transition, which is a second order transition, the velocity goes as the deviation from that critical point to the power of 3 by 2. Okay, 3 by 2, numerically 3 by 2. Yeah. On the boundary conditions, that's right, yeah. So, so if you're looking at scales which are much larger than the mesh size, much smaller than the mesh size, then it appears as boundary conditions. If you're looking at scales much larger than the mesh size, then it appears as quenched random anchoring variables. So then you integrate that out, and basically they appear as parameters in the equation.
Right now, none of what you're doing has quenched random at this level.
At this level, nothing, none of this. Yeah, okay. So, yeah, I'll skip this. Okay, so there are two scales in this Aster of lattice. One is the distance between these Asters, which is set by the contractility, which is set by this term here, the ratio of this term to that. And the other is the size of the Aster, which is set by the ratio of the diffusion coefficient to the advection, okay, or the Peclet number. Where are we now? I have to go back, down. Okay.
But all of this is for the case where the sign of one of those coefficients
is one sign, yeah.
Your zeta has one particular sign.
Yeah, yeah. So this is for zeta being positive. For zeta negative, is the uniformly oriented phase is stable. Okay, so this zeta being positive is the appropriate sign for active myosin contractility, as shown by Tani, Liverpool, and Christina. Okay. Okay. So now those are the steady states. You had a moving phase at low contractility, and at high contractility, it locked into a stationary Aster lattice phase. And the parameters of the lattice are set by contractility and various parameters that enter into the equations of the orientation. Now we turn on the noise and ask for what is the dynamics in this steady state. And that's quite interesting. At low values of the active noise, you find that, so here we are, at low values of active noise, these Asters, which form the square lattice, just do phonon jiggling. Okay? So you can compute the structure factor and show that there's a Debye bipolar reduction coming from this active temperature. As you increase the active temperature further, the Asters remodel. Okay? An Aster which was there for some time suddenly breaks up and reforms somewhere else. So this is what is plotted here. If I look at the Aster strength, then it is 1 and 0, and it's stochastic. Okay? And you can calculate what is the distribution of the lifetimes of this aster, and you find that in region 2, it has a power law distribution. As you increase the active temperature, it goes over to an exponential distribution. Okay? So these two phases are phases where the asters form, they break up, they reform somewhere else. Okay? And in this phase, in region 3, this remodeling happens very, very fast.
Well, it's really a power law, meaning if you do different system sizes, it doesn't seem to cut off.
It doesn't seem to cut off. Yeah, yeah. Okay. Now these regions 1, 2, 3 are surprisingly associated with interesting structural phase transitions. Here we have computed a variety of order parameters. So this is the bond orientation order parameter. Remember, it's a square lattice. So the appropriate thing to think about is the tetratic order parameter, and that's what we compute here. At temperatures, so this is the region 2, and this is region 3. Okay, in region 2, the psi 4 is high. This bond orientational order is high, and then crosses over to a low value in region 3. Similarly, in region 2, psi 2 is low, and then crosses over to a high value in region 3. Okay? Here is the net orientation of the filaments, which is low in regions in this region 2, and in fact is 0 in region 1. But in region 3, when you have the exponential remodeling phase, it's quite high. Okay? So this has given rise to a tentative phase diagram. The jiggles that you see are not coming from any calculation, but because my hand was jiggling. But if I plot the contractility versus active temperature, then this seems to be the set of phases that we observe. It's a really complicated phase diagram. There might be more phases. So what I showed you is when we went from the aster crystal phase, the lattice phase, to a tetratic phase, to a bond nematic, to a polar phase, and then finally to an isotropic phase. Okay? We need to do more work to actually get correct boundaries, but we have a few of these things in place, as I showed you here. Okay.
What is the Bujam?
Yeah, it was named after that. Yeah. For a Bujam, you need a boundary. No, but this is not a single. Remember, this is a collection of Bujams. So you don't, no. But they're not singular. They're not singular, so that I can show you, we can blow it up later on. Okay, so now, what I described so far is the dynamics of this actin below with all these interesting phases, and they form these asters which make and break and so on. How do they now influence molecules which sit on the cell surface? So we'll define 3 classes of cell surface molecules. This is inspired from discussions of how particles behave when they're inserted in a fluid. Okay? So we'll call them inert particles, passive particles, or active particles. Inert particles are those which do not interact with this dynamic actin. Example being short chain lipids like NBD sphingomyelin. Passive cell surface molecules are those which interact with dynamic actin but do not remodel the actin themselves. An example being the Gpi-Anchored Proteins that we've been discussing. Active cell surface molecules are those which interact with dynamic actin and also remodel it. And there are lots of interesting examples, integrin receptor being one of them. So what I'll talk about today will be these 2 classes of cell surface molecules. Okay? There's ongoing work in Satyajit Mayer's lab, where we are looking at the dynamics of integrins and other signaling receptors. Okay, so here is what we imagine it to be. If you have an actin filament and a cell surface molecule, the cell surface molecule can either bind onto the actin filament with the rate k on or unbind with the rate k off. When it binds, it's advected along the filament. When it's unbound, it just diffuses. Okay? And there's some thermal noise there. These phi's here are rates, stochastic on-off rates. If it's 1, then it's on. If it's 0, then it's off. Okay? So now you can imagine that if your local configuration of the polar actin has this aster form, then by this process it will drag molecules to the cores of the aster. And so you can get a clustering of molecules, not arising from protein-protein interactions, but because of its coupling to this active actin below. Okay, so I can skip that. Yeah. So now this theory allows us to calculate various things, and so we can compare the results of the theory with experiments which we had done in these papers. So this is the single point distribution of the local density minus the average density, plotted against the local density. You find when it's an inert particle, that is, it does not interact with the dynamic actin, the log-log plot shows a parabola, which means that the distribution is a Gaussian. Okay? However, when the particles are interacting with actin, like passive cell surface molecules, then you get these very significant exponential tails at either end. This is the experiment, which is obtained by doing local FET anisotropy measurements on the cell surface. This here in blue is short chain lipid molecules, which do not interact with actin below, and they have this Gaussian distribution in the log-log plot. Okay? However, Gpi-anchored proteins, you see these significant tails. When you get rid of actin on the cell surface, or get rid of or use blebistatin, which is a way of stopping contractility, then you find that these exponential tails get suppressed. Okay? Now, as I said, the asters make and they break and they form somewhere else. If they are housing the cell surface molecules, then the cell surface molecules will form clusters, then they'll be dispersed and they'll form somewhere else and so on. So there's a rate of fragmentation and aggregation of these Gpi-anchored proteins. So that's what is plotted here. The log of the aggregation to fragmentation rate on the y-axis versus 1 over the active temperature on the x-axis. And it has this non-Arrhenius behavior. At low values of active temperature, there's not much of breaking and making because the asters don't break too much. Okay? Whereas at high active temperature, the asters break and reform a lot, and therefore there's a lot of aggregation fragmentation. This is what we observe on the cell surface. This is again log of the aggregation fragmentation rate versus 1 over the real temperature, and it shows this peculiar behavior. Temperatures below 24 degrees, which coincides with a low actomyosin contractile activity, you have very little fragmentation aggregation of the GPI clusters. At temperatures between 24 and 37 degrees, there's a very high fragmentation aggregation rate. Okay? This is the other feature that we see. If you look at the fraction of proteins in nanoclusters, it is independent of the total number of cell surface molecules over a large range. This is what we see in theory. In experiment, we just see the same thing. Different cells express different amounts of the cell surface molecule, but independent of that, the fraction of molecules on the cell surface is the same. Okay. So now let's look at a new experiment. Let's look at a prediction that comes from the theory and see if we can test it experimentally. And from our theory, if we look at the simulations, we find that I locally look at this region, I find a lot of, let's say, nanoclusters here. As a function of time, these nanoclusters disappear and reform somewhere else. So it seems clear that there's a tremendous amount of fluctuations of the local density of these passive molecules. Okay? So the question now is, can we compute these fluctuations in the number of passive molecules, and can we experimentally measure these fluctuations in the number of the Gpi-anchored proteins on the cell surface? So what we have to do is, this is the root mean square fluctuations of the number of the passive molecules, and that's equal to this quantity, the cube tending to 0 limit of the structure factor. Okay? And the structure factor, the density-density correlation, equal time density-density correlation function, can be computed from this theory. In fact, we didn't have to do much of the computation. The computation was done earlier by Toner and Tu. And what they showed was that density-density correlation function has this following scaling form. Okay? So it goes as q to the power of minus 2 zeta, where zeta in 2 dimensions is 3 by 5, an exact result in 2 dimensions. This formula translates to the fact that the root mean square fluctuations of the number scales as the number to the power of 4 by 5 and not square root n, as you would expect from a Brownian particle. Okay, so let's see what we see in experiments. In experiments, what we do is you take certain windows of different size on the cell surface, you carve out different windows, and you have fluorescently labeled these Gpi-anchored proteins, so you can look at the time series of the fluorescence intensity in each window. From the time series of the intensity, you can compute the mean intensity and the standard deviation. And then you can plot the standard deviation versus the mean of the intensity. That should be clearly related to the standard deviation versus mean of the number. Okay? And this is what we get. If we do it for an inert particle, like short chain lipids, etc., you find that this, the root mean square n is proportional to square root n, okay? What you would expect from a Brownian particle. However, for large values of n for the Gpi-anchored proteins, you find a larger scaling, and this is the line n to the 4 by 5, which is 0.8, okay? Which is significantly larger than 0.5 for large n.
To the eye, the red dots don't seem to be parallel to the red line to guide the eye.
This side?
No? It seems to be that.
So it may not be exactly 0.8, but
Yeah,
it's
better than bigger than 0.5.
It's bigger than 0.5. So I don't. Yeah, exactly.
That's
it. I won't stake my life on 0.8, but it's certainly different from 0.5. That's what I'd like to say. And
sorry. Okay. Yeah. I'm sorry I started this.
Okay. So now what we can do, if this is true, then what we can do is we can sort of remove the activity of the cell surface by a cocktail of chemicals, lataclin, jas, and blebistatin, and reduce the influence of the cortical activity of actin on these Gpi-anchored proteins. And what we should see, of course, you can't remove it entirely, but what we should see, and what we do see, is that the crossover from square root n at low n to n to the power of 0.8 happens at much larger values of n. Okay, and that's consistent with what we would expect from this behavior. So that's what I won't go through it, but that's what we see here. Okay. Yeah. Okay. So now we just calculated the equal time density correlation function of Gpi-anchored proteins. You can also calculate the unequal time density correlation function of the Gpi-anchored proteins or of passive molecules. And this is the unequal time density correlation function is related to the diffusion coefficient. Okay, and the diffusion coefficient of cell surface molecules can be measured by FCS. So here are preliminary results, and this is not a. So this is the time it takes to diffuse over a region over a confocal volume versus temperature. Okay, this is for Gpi-anchored proteins, and this is for Gpi-anchored proteins on blebs. That is where the cell surface has been detached from the cortical actin. Okay, here you find that it has a temperature dependence consistent with Stokes-Einstein, whereas here on the live cell surface where the cortical actin is talking to the cell membrane, you find that it's independent of temperature, and this sort of suggests that maybe the fluctuations that give rise to diffusive behavior of the Gpi-anchored proteins are arising not from thermal fluctuations but from active fluctuations. Okay, but a lot more work needs to be done on this. We are now currently analyzing these set of experiments quantitatively with the theory that I just talked about. Okay, so in summary, this is what we, I mean, this is not the summary of the talk, summary of the section, is that for inert particles, like so, short chain lipids, the single particle distribution of the density is a Gaussian. The variance of n is square root n. The diffusion coefficient extracted from FCS experiments is linearly dependent on temperature. For passive cell surface particles like Gpi-anchored proteins, RAS molecules, etc., the single particle distribution as a function of density, local density, has these significant exponential tails. It has a variance which goes as n to the power of 0.8, not larger than 0.5. It has a diffusion coefficient, which again you can extract from FCS, which seems to be independent of temperature. Okay, how much time do you have?
It is 4:40. For full time, you have 20 more minutes.
Oh, okay.
Including questions.
Yeah. Okay. So now having studied as you can see, that these asters are an interesting way of concentrating molecules to the cores of these asters. And so you can imagine that you could concentrate two molecules and they could engage in chemical reaction. So by having a bed of asters which locally catch molecules and drive them to the cores, you can tremendously enhance the rates of chemical reactions. So this is work done by Abhishek Chaudhary and Baswati, who was here. And the idea was to study a whole variety of chemical reactions and see whether these dynamic asters that are formed indeed gives rise to an enhancement of chemical reactions. Okay. So this is what we do. This is studied by a Brownian dynamic simulation, and you sprinkle the 2D surface with asters. These asters have a certain lifetime. There's an exponential distribution of lifetime of these asters, and with a mean in the distribution, which depends on the local active temperature. Okay. So the molecules that the molecules that move around in this 2D surface can bind to these filaments and get dragged, can be driven to the cores of these asters. If they are not caught by these asters, they are happily diffusing around. Okay, so that's the dynamics. We've looked at a whole lot of chemical reactions, second order and higher order chemical reactions that I show here. But let me just give you a generic picture that we get. Okay. One interesting thing here is remember there are two terms in the particle current. One is the diffusion term, and the other is an advection term. How fast molecules which bind to actin are driven to the cores of these actin. So that gives rise to a Peclet number. One can define a Peclet number, which is the rate at which advection happens to the rate at which diffusion happens. Okay? When the local Peclet number is beyond a critical value, let's say around 10, then you find that there's a tremendous gain in the rates of chemical reactions. Okay? And this is drawn for a particular kind of chemical reaction, where there's a gain at around 10, there's a gain from being less efficient than diffusion to 10 times more efficient than diffusion. Okay. So as I said, driving these molecules onto the cores of asters enhances the rates of chemical reaction. Now this is an interesting thing. This has to do with not only the ability of asters to drive molecules to the cores, but also the fact that these asters are not stationary, but make and break. Okay? And this says that there is a particular time scale associated, that is, when the time it takes for molecules to be driven to the cores of the asters is exactly the same as the time it takes for the asters to fragment. Then there's a resonance condition is met, and you get a very high enhancement in the chemical reaction. Okay? So this is an analog of stochastic resonance, but it's coming from activity. You're equating two time scales, one having to do with the advection of molecules to the cores of the aster, and the other to do with the breakup of these asters. Okay, so there's a phase diagram that one can draw in the rate of advection versus the rate of fragmentation of these asters. And you find that in this region in red, there's a tremendous gain in the reaction rates, and in this region there's a suppression of the reactions. Yeah. Which is 3 regions? In the first one? Yeah, this one. No. So this is the phase boundary. Yeah, this is a line where I meet the stochastic resonance condition. So this is that. So I'm just drawing a line there to show that it roughly this corresponds to the maximum of the chemical reaction as you go along this. Okay. Okay. So this is the bullseye pattern that I talked about. So I'll now very quickly go through this because we don't have detailed calculations as yet. We just started these calculations, and this calculation is done by Abhishek Chaudhary in collaboration with Ron Whale and G2. And the idea here, so this is following some work by Ron Whale and Mike Dustin's group, where they found that on the T cell surface, you find this familiar bullseye pattern, okay, where T cell receptors are clustered in between, in the core there, and ICAM receptors form this halo, okay. And what they find is when they try to picture the actin below, this is how the actin looks like. Oops, you can't see it. That is where it is. But what happens is that you find the actin forms a halo around that, okay. This picture would have shown you lines of actin streaking across like that, okay. So the idea here that they had was that these molecules are being driven towards the interior of this actin aster. They didn't call it aster, but actin aster, to form patterns like this. Okay. So what we did was to say that let's now imagine that these I'll just call them A and B, okay? T cell receptors and ICAM1 receptors. A and B are molecules which sit on the cell surface, and they not only couple to actin, but they also interact with each other. Okay? They have a weak attraction to each other. And I'll just model that by some Morse potential like so. Okay? So alpha beta alpha is either A or B. Okay? And that's the form that I would take. And what you find here is that depending on the magnitudes of the depth of the potential, how strong AA interacts compared to BB, you find an interesting set of patterns that you get. The bullseye pattern forms when the AA interaction is much stronger than the BB interaction or the AB interaction, and both of them bind very strongly to actin. Okay? But you also get a whole lot of different patterns, and there are examples of each of these in this whole T cell signaling cascade. Okay? This is true for T cell receptors, TCR and ICAM, but there's another molecule called LAT, which seems to have this kind of architecture. Okay. Which one? So think of that as mod, yeah. So in this case, it's positive, yeah. But think of these as mods, yeah. Okay, so this tells you the well depth, yeah. So V alpha beta is the well depth of that. Okay. Okay. Now I don't know whether I need to go through this. Maybe. Okay. Okay. So maybe I'll just end the last part of the talk with the formation of cortical actin rings on spherical and cylindrical cells. This work is done by Pragya, who's sitting there. Unfortunately, she didn't give me her calculations, but I'll just tell you what we have done. Okay. So what is done is the following. So these are experiments done in Mohan Balasubramanian's lab at Singapore, and what they do is the following. These are fission yeast cells. Fission yeasts are generally cylindrical shape because of the existence of a very strong cell wall. There are two sets of experiments that they do. One is that they disrupt the cell wall, and therefore, because of turgor pressure, the cylindrical cell forms a big spherical cell like that. Okay? Or they mutate a certain chemical in the wild type cell so that the cortical ring that forms, an actin ring that forms, can't stabilize anywhere. Okay, when they do, okay. So now in the spherical cells, so this is what happens in the sphere when it forms a sphere. Okay, so what is labeled in green is actually myosin, okay, which follows actin. And what you see here is that there's an actin ring that is formed, but the actin ring can never stabilize. Okay, it just slips. That's in the spherical cell. Here, because of mutating that particular chemical, mutating the particular chemical. Okay, it doesn't work. But by mutating the particular chemical, the cortical ring doesn't form in the cylindrical part of it, but forms at the end caps. And then forms at the end caps again because of the curvature, it slides down. Okay, so these two are curvature-induced effects. The slipping or the instability of the cortical actin arises because of its coupling to the curvature of the membrane. Whenever it finds itself on a spherically curved membrane, then it slips However, when it finds itself on a cylinder, it diffuses, okay, unless it's held by other anchoring agents. So what Pragya was able to do was to use the same kind of theory that we just talked about, but write it for a cylindrical surface and a spherical surface. Okay, so do the same calculation, but write it for filaments sitting on a sphere or on a cylinder. And from these set of equations, you can actually calculate the contractile velocity or the slipping velocity and compare it with experiments. Okay, and again, unfortunately, I don't have the data here, but our work is quite encouraging, and we find that the dynamics that I talked about gives rise to a natural explanation of how contractile forces can make rings of actin slip because of curvature. Okay. Right. So, okay. So in this particular case, in this particular case, it doesn't squeeze the cell. Okay. However, often you find that the cell here has a little funny outgrowth. Okay. So it has, instead of being entirely a sphere, it has a sphere with a little nose there. Then what happens is that occasionally the ring forms there, and once it forms there, it gets stable and then starts squeezing. Even here in this cylindrical case, if it finds itself in the middle, it'll squeeze it. However, when it localizes at the end caps, then it doesn't squeeze it. Okay, so I'll end the presentation here with just giving a list of my collaborators. All the work that I talked about was done in extremely close collaboration with Satyajit Mayer, who is in NCBS, and his group. So these are the set of people who did the fluorescence-based experiments that I talked about. The actihydrodynamics theory, largely the initial part of the work was done by Kripa, who's just finished her thesis, and is being continued by Pragya and Roy Shlomowitz, who is at Weizmann. The work on the chemical reactions and the bullseye patterning was done by Abhishek Chaudhary and Baswati Bhattacharya. Abhishek is now in Oxford doing a postdoc, and Baswati has just finished a PhD. These are the experimentalists who did the work on the contractile slipping of cortical actin in these cells. And I've been having very fruitful discussions with Sriram here and with John Toner, and our work has been funded by these groups. Thank you.
Why does the Aster lattice have a square symmetry?
So that's a good question. Okay, so I'll have to. The square is the following. Okay, so there's an. Okay, so you can. If I just look at these terms in the right-hand side of the equation, yeah, then they, in principle, can be derived from an energy. It's inappropriate to talk of energy in this active situation. In principle, these terms can be derived from what is the energy cost in distorting the orientation of n. Okay? If you use that part of the energy, then you find that the square lattice does better than a triangular lattice. Okay? Now that's the result of a calculation. I don't know of any deeper reason than that.
Thank you.
Other questions?
Yeah, so these Asters, they don't have a, do they have a directionality?
Yeah, so these are polar filaments. So they are pointing inwards.
They are always pointing inwards?
They're always, so, okay. So in general, if you take a single aster, you can show that the inward pointing aster is stable and is stabilized by advection coming in and diffusion trying to pull it out. Okay? If you now make the asters point in the outside direction, then this advective term, this term will make the arrows go to infinity. Okay? So the outward pointing asters are unstable. However, if I have a collection, an array of asters, then you can ask what happens in between. And in between, you have either local splay-like things or anti-asters.
Okay. So does that explain the square lattice better?
Better meaning? So I know
without referring to the energy.
Sorry?
Without referring to the energy. Just can you do that can you do it
No,
in
sir,
a triangular?
if you have a triangular lattice, you can also put anti-Asters, yeah. Referring to the following thing, that if you have disks with an arrow, and the arrow, and let's say Ising arrows, and you have energy for the density field and the energy cost for arrows, then often it's better to go into a square lattice than into a triangular lattice, right? If you have antiferromagnetic order of the.
None of this depends on the K1, K2 ratio.
Yeah. So if you change the K1, K2 ratio, so if you change, so that's K1, K2, K1,
Back.
K1, K2. Yeah. Okay. So most of the work has been done where K1 is roughly the same as K2, but we have actually, as you see here, we have changed. So delta K is the difference in K1. However, if you make the difference large, then instead of getting Asters, inward pointing Asters, you get inward pointing spirals. So you can get spirals by both making the difference between K1 and K2 large, and by changing lambda. I don't understand why changing lambda gives you spirals. But that's good. So lambda, there's no problem. But you can't change the sign of K1 and K2.
That would be bad. Okay, if there's no other questions, let's thank Madan and
Thanks, Sriram, for chairing this session. And a whole another week to go. So I think for discussion. So, but anyway, we come to the end of the conference program. It's been a lot of nice talks and interactions, and I won't spend too much time with the concluding session, but it's customary to thank all the people who sort of helped with making this possible. So let me take this opportunity. We've had a little bit of a rough time putting this program together because the key organizers were scattered across the globe, and we had no secretary for long stretches of time. But so let me start by thanking actually my students who sort of came into the rescue at various points. Vishwas, Aparna, Shiladitya, Shibu, and Momita. So for work beyond the call of duty. And Supriya, who has been helping us as a conference secretary for the last month or so. And Sachin, who's our public relations assistant, who helps in all these activities, our administrative officer, Jai Chandra, the staff in the conference hall, Mune Gowda, Peer, I forgot his name, but that guy. And the JNC staff and administrative office in general. That's sort of the organizational part. A number of people have sort of helped with ideas and suggestions, who members of the organizing and advisory committees. I want to thank them, starting also with Surajit, who's been the co-convener of this event. And I want to thank all the speakers at this meeting for making it out here and telling us interesting things, and all the participants.